There we go. Okay, cool. So we're going to talk about a rather cool article, which is unfortunately insanely complicated, but it's really, really cool. So we're going to talk about it, and I'll kind of simplify the important parts of it. Basically, so the title is Characterization of Antibiotic Resistomes by Reprogrammed Bacteriophage-Enabled Functional Metagenomics in Clinical Trials, Clinical Strains. That's a really big mouthful. What they're basically doing is they're saying, hey, we'd like to figure out how we can quickly identify antibiotic resistant sections of genomes and be able to rapidly develop and test them in different model organisms in a convenient way that can be done kind of in an automated process. And part of the problem with this is that most of our work with this is in something like E. coli, which is fine, but some of those don't work very, very well, and we just need a better model. And so the other issue is, how do we get these genes into them? Well, we can do these genes at an incredibly slow pace with regular old transformation or electroporation. So they do a lot of these with electroporation. Now the results for electroporation, they're okay, but the diversity of results is really, really, really low, which is not great. But with phages infections, it's actually way, way better. So what they did, they go, hey, we've got some samples from clinical settings. So they took clinical settings, soil, and some other ones. They grabbed a whole bunch of genomes that they were like, hey, these genes are antibiotic resistant genes. We're gonna plug them into these phages, and we're going to use these phages to infect other types of bacteria. Now, they did end up running into a problem because phages are super, super, super specific to only one type of species. In fact, even more so, they can only be really good at infecting one substrain of that species or one serotype of that species, which makes it really obnoxious. So these guys are like, okay, we can fix that problem. Oops. So what they did is they were like, hey, no problem. We will stick all of these phage thingies in here. We will stick all these things in here. We will then also stick in a plasmid that has this mutatable, easily mutatable T-phage binding region. So it allows the phage, the part that allows the phage, the leg part that binds the bacteria. And we're going to generate this in E. coli, then infect these E. coli with the genomes that we want, with a bunch of phages that actually have the portion that makes the tail and leg cut out. So they cut out that gene from the phage genome. So the phage infects, and they have to be able to incorporate those genes, these randomized ones, and be able to be successfully infective. So they just kind of get those randomized tails put on automatically by the bacteria, producing the gene from the bacteria's plasmid. And so they also introduce a series of small mutations and basically are able to kind of brute force which phages are able to infect which bacteria the best. And so you can see here, they had a whole bunch of things. So they were like, okay, look, here's different types of tails. So they used T-phage. They used this one that's primarily infects Salmonella typhimurium, and this other one that I can't remember exactly what it targets, but you don't need to worry about it too much because that one didn't work as well. But basically they were able to show, hey, these are able to infect a completely different species of E. coli and a completely different bacteria altogether, and this one much better. And you can see the difference between here and here isn't huge, but the difference from here to here, and especially from here to here, is massive, because remember, this is on a logarithmic scale, so it's much bigger every time you go up. And similarly for this one as well. So they were able to get way better infection rates of the phages with these recombined tails. So they were basically setting up the system, and they were saying, hey, let's get a system where we can really accurately, really quickly figure out a whole bunch of phages that can affect a whole bunch of different types of bacteria, which is important, which we'll explain why in just a second. And we're going to use them to then figure out where the antibiotic resistant genes are and if they work in other bacteria. Because one of the big issues is that these antibiotic resistomes, they call them resistomes rather than resistance genes, is because it's actually not one gene for a lot of these things. A lot of them are actually a collection of genes, and we don't actually know what those connections are. We just happen to know that there might be one gene that's associated with primarily, but if you stick that one gene in a new bacteria, that new bacteria does not have antibiotic resistance to that antibiotic. It just doesn't work. And so it makes it really hard to kind of figure out how it's working and interacting together. That's the reason why we need to be able to test this in multiple organisms. So they did this whole thing. They tried it the first time, right? With they did it with a substrain of Klebsiella pneumonia, and they were able to figure out, okay, yes, all 10 of these resistance genes that they had, or all of these resistance genes that work in Klebsiella pneumonia, worked in Klebsiella pneumonia also that didn't have those resistant genes. So it worked to the species correctly. Good. So the system works. And then they did it with E. coli and a bunch of other hosts. So they performed the exact same experiment, but this time they were able to show that, yeah, it does work in E. coli, but if we didn't do it with this special system where we had all these phages that were able to infect brand new hosts, we wouldn't have been able to find what is almost 30, I think it was 37% of these antibiotic resistant genes. They wouldn't have been able to find them because they wouldn't work in E. coli. They only work in those other hosts, which is crazy. That's a huge amount. So they were able to show that a lot of this stuff here, and I just like this chart really quickly because it shows that the genes that are involved in this tend to be related to each other, especially if your classes of antibiotics. So if you guys know anything about antibiotics, you know that they're generally broken into these major classes. So you've got things like beta-lactamases or sulfonamides or macrolides and things like that. So there's these major classes. And a lot of our new antibiotics tend to be variations within that class, so we make a small change to the molecule. And so they wanted to point out specifically, their really important one, which was this guy, Ceph, which is their 5th generation cephalosporin that was recently created. So it's brand new, FDA just cleared it. So that means the likelihood of it having resistance developed against it already specifically is really low. But they were able to find that a lot of organisms, right, that have resistance to similar antibiotics within that same class confer that resistance to that guy right there, which is not good, but it does show and give you kind of an opportunity to say, hey, which antibiotics are going to work really well against these organisms, and allows you to test these things out a lot sooner and kind of get a broader picture of where antibiotic resistance comes from and what kind of modifications you need to make that help enable better susceptibility in those bacteria. So basically they had a really big success in this, in that they were able to create the platform where they put in a whole bunch of phages that were able to infect a whole bunch of different species that they normally shouldn't. And they got less great results in terms of being able to kind of really dig down into these antibiotic resistance genes, but they will be able to do that later on. So they were just kind of like, hey, here's our first volley. It gave us some information, not great, but the system, the platform works. So what do you take away from this? All you need to take away from this is A, if you are an antibiotic in the same class and something's already resistant to the other antibiotics within that class, chances are, even if they've never seen that antibiotic before, they're probably going to have some degree of resistance to it. Also, the way we got these things to infect is by shuffling up the tails with random mutations and then exposing it to a whole bunch of other bacterial hosts that we normally couldn't. And the reason why that's important is because some antibiotic genes only work with discrete parts of the genome that are not present in every single organism. So if you got it from Salmonella, it's like Salmonella typhimurium, okay? And you were like, hey, let's put it in Salmonella sonnei and see if it works. There was no way to do that because the phage didn't exist. But now there is, because it would work much better in Salmonella sonnei than it would work in E. coli or Bacillus subtilis, because those are very disparate from each other and they don't have the right gene setup that antibiotic resist them. So all you have to take away from this is we were able to shuffle the tails to create new randomized mutations that allowed it to infect better. Okay, any questions on that? I know that was really complicated, and trust me, if you read the paper, it's even more complicated than I made it sound, because they used neural pathway mapping and other stuff, but It's like, I think it's like a 30-page paper, so. Okay, so how do you not fail? Very important. Generally speaking, you want to review the previous lab PowerPoints, panic moments before the practical, write down C for every single answer, and then don't put your name on paper to delay the inevitable. That works really well for some people, but a much better method is to actually go and review the previous lab lectures, which is important. Know the steps in each test we do, so anytime we do a test, be like, hey, I know what thing we're doing in which order and why we're doing it. Also know the reagents that we use and why. So for example, for gram staining, like why do we use each type of stain and what is its purpose? Or what is the purpose of the decolorizer? And then know what the results should look like, especially for things like selective and differential plates, because that's the most recent one. That also has like a large chunk of information, and a lot of people tend to be like overwhelmed by how much information came from that. So that's one that's really important to know. How this is going to break down. On Monday, for you guys will do a skill check, 2 skill checks. One, you guys will get a plate with a mixed broth. The mixed broth is not going to be the same bacteria that we used last time. It's going to be 2 different bacterium. And you guys will mix it up and streak it on an isolation plate and try to get 2 of each colony. I will do this beforehand to verify that it works. I'll even show you guys a plate that has it mixed and ready to go for you guys. But you basically just have to get 1 of each type of colony isolated on that plate, so you guys will stick it in the incubator and your TAs will grade it later. The next thing is that you guys will have to do a pipetting a volume. So your TA will call you guys up by a group of 4 and have you guys draw a random number volume. You guys have to choose the right pipette, set it to the right volume, show the TA, and they'll tell you if it's right or not. If it's not right, they'll have you go again so that you can actually get the pipetting part right. Then you'll have to sterilely take a tip, suck up liquid from one container and put it in another container, and the TA will measure that against a known volume already. Really important part here though, the sterility part that trips people up. The most important part with the sterility part is A, when you guys have the pipette tip, once you take the lid off the tip box, you push down to get the tip on there, pull it out, you want to put the lid back on, because if you leave the lid off, now you're just contaminating all the tips with the junk from the air and from you breathing on them. That's bad, so put the lid back on. Then you want to make sure to be very careful not to touch the tip against anything other than the liquid that you're going into and the test tube that you're going to transfer the liquid into. So be very careful with that. The last part, which is also very important for sterility, after you pipette it, what's the last thing you do? What was that? Eject the tip, yeah. So we want to dispose of the tip properly and make sure to close the tip box, but make sure everything's clean and sterile. So that's the most important thing. Making sure you know how those volumes work, making sure you know how to pipette correctly, making sure your volumes are kind of like in a good spot. Okay, so there's that. Let's test a couple of questions. Okay. Actually, this is the wrong slide, and I don't have the right one for you guys. Okay, so we're gonna skip down here for a second. Because you guys are not doing acid fast. So you guys don't have to know the acid fast stuff, you don't have to know the endospore stuff. This is the stuff that you guys have to know. For you guys, it's lab safety, basic microscopy, so being able to focus a microscope, basic handling and culture techniques, making sure you're pipetting properly, gram staining, negative staining, transformation, pure culture isolation, viable plate count, oxygen requirements, water testing, selective and differential media, nitrogen cycle, and lastly, for the bonus questions, those are going to be your cool science things week. Bonus questions are about, I usually make between 5 and 7 questions. The total exam is only about 70 questions in total, plus the 7 bonus. So if you do all the bonus, you can change your whole grade by a letter grade. So, you know, keep that in mind. I'm very generous with those. Yes? Yeah, there's a practical portion. So the first thing you do is you do the plate isolation, and then you're going to do the pipetting. And then on Wednesday for you guys will do the actual practical. And I'll show you guys what the practical is set up as in just a second here. But basically these are the topics that you guys should know. Yes?
You said 7 questions?
The bonus. The bonus. There's
How many questions like on the actual practice?
around 70. So 70 plus the 7 bonus, so 77 in total. You don't have to do the bonus if you don't want to. Feel free to ignore it, but yeah.
The bonus, since it's the full science thing of the week, is it questions over,
The things that I talked about.
yes, like, you know, you had like listed the long paper, should we read the paper?
No, it's just over the things I talk about. I wouldn't make you guys read the papers because the papers are 10 times more complicated than what I present them as. Most of them are. Good question. Yes.
Is the exam multiple choice or fill in the blank?
Very good. The questions are fill in the blank. They're generally 1 or 2 word answers. So I don't need like a sentence out of you guys, but 1 or 2 word answers. So you guys have to know these things kind of off the top of your head. I'm not going to take off for spelling because I don't like to spell without a computer. As long as you get it mostly right to the word, then that's totally fine. But, you know, keep that in mind. Good question. Yes?
Assuming there's going to be some degree of application questions on the exam, how are we supposed to go about the free response or the without multiple choice? In the sense of like, well, how do we know what you're going to look for or the quantity of words you want to put on the...
1 or 2 words, pretty much maximum. So we'll give you guys an example. Here, this is one that's functional for you guys. What kind of plate is this? Okay, great. That's already, you guys are already way ahead of the curve. Okay, great. All right. Oops, I already showed you guys that. Okay, what kind of organismal growth does it support? Fastidious. What does fastidious mean? Something that's fast, something that's acid fast? No. If an organism's fastidious, what does that mean? Yeah, it needs an enriched specific media. So it has certain requirements that it needs to be able to grow effectively. So a great example of this is blood agar, because a lot of organisms need the nutrients on blood agar to function properly. And so they'll grow really well on blood agar. They might grow on some other medias, but they really prefer blood agar. Great. Okay, this specific plate. What is the result for this plate? Gamma, which is also known as non-hemolytic. Right. Very good. Okay. And lastly, what makes the plate opaque? Wait, say that again, guys? Red blood cells, yes. So red blood cells are the things that make it opaque. So unlike a lot of the other media that we use in the lab, if you hold it up to the light, you can see right through it. This one's opaque. You can't see through it because of the red blood cells. All right. And guys, I'll give you guys another test question because this one was obviously, because we're not doing acid fast, you guys don't have to worry about it. But I'll give you that in a second. What we're going to do is we're going to talk about the format. So it's going to be a short battle royale where you have to compete against your lab partners, and whoever comes out on top gets an A. No, that's not what we're going to do. What we're going to instead do is have 25 stations in total, 25 or 24, and basically it's where you're sitting at your seats. We're all going to sit down where we normally sit at the beginning of the day. You guys will get a set amount of time at the start. It's about 2 minutes per station, and you gotta get up and move once the timer goes off. So you have 2 minutes at station 1, then you move over to station 2, which is seat number 2. And you just kind of go down the line until you get to the very end. Once everybody's done, we will give you guys time at the end to go back and look at stations for about a minute or so. Don't hog one particular station. But you know, if you can get it, you can get it. And if you can't, you can't. If more than one people are at a station, try to form a line. Don't crowd up on the same thing because it makes it kind of look like you're cheating, which is bad. And we want to respect the honor code for Baylor's, making sure that we're not cheating or getting the illusion of cheating. Because I know you guys wouldn't, because you guys are trustworthy students, right? Yes? I believe that. I actually do. Now, once you go through all the stations, you guys will have time at the end to turn them in. Once you guys are done, you guys will make sure to put your name on them. Turn them into your TA. They will grade them and add up all the points, including this practical skills check they'll put on top of that. Now, one of the stations will be the bonus question station. Each of these stations will have between 3 and 4 questions on each of them. And it might be something like that one slide I showed you guys with the red blood cells, the red blood agar plate. And you might go, okay, well now I know what that is. I know what kind of media it is. I kind of know what this growth is. What is this organism? growing what is the result. And I really care a lot about results for this. So like, for example, if I were to show you guys a microscope with a slide on it, and it has dark purple bacteria in it that are circles in little patterns of 4, what would you call that? What's the gram result for that? Gram positive, good. What's the shape? Cosi, yeah. So shape can be cosi, bacilli, sporuli, a couple other weird ones which we don't have in lab, so don't worry about them. And then the arrangement. The arrangement's kind of a tricky one. What's the arrangement for little patterns of 4? Okay, does anybody know? Raise your hand and we'll hear one person. No? Tricky one. Tetrad. Did you know that? I saw your hand just as I said it, yeah. Okay. There are a variety of other arrangements. You should be roughly familiar with them. The really common one is random, where there's no arrangement, it's just kind of all spread out. It could also be strepto, which is in a line, that could be the cosi or bacilli. Staphylo, which is little clusters of grapes, which only applies to the cosi ones. And then you can also have palisade, which are kind of like stacked on top of each other. They're bacilli, so they kind of form like a little palisade line, like a white picket fence or a fort palisade, that kind of thing. That would be another one. Okay, so that would be an example for that one. If I give you a different one that's a little bit more tricky, one of the ones that people tend to forget are things like figuring out the MPN chart. If you guys don't remember how to use the MPN chart, that's a really good one to make sure you guys know how to use. So with the MPN chart, there are 3 volumes that we put in those phenol red broth tubes. What are those 3 volumes? Does anybody know? 10, 1, and 0.1. Great. Now, which of those tubes is the double strength phenol red broths? Okay, why is it the 10 mil? Why does it have to be the 10 mil? This is a large volume, very good. So because there's so much extra water, if we just use the single strength, it would be so dilute that the pH would never change. So that's why we have to do extra lactose in that one. Very good. Perfect. Okay. That was a little bit more of a hard question that I wouldn't ask, but I'm glad you guys know now. Okay. Okay. So with that, we can check if there's any questions. Just use Google. No, I'm kidding. That is just a joke. Do you guys have any other questions or things that you'd like me to go over? There is a little study guide that's kind of fill in the blank. Some of those things aren't on the review, like antibiotic. We shuffled the labs around, so antibiotics is not on the review for you guys, but it's got a bunch of other ones. Yes, in the back.
Just to clarify, we're not responsible for the labs that we missed because of the snow.
Correct. So I cut out the acid fast and the endospore. But you guys are responsible for gram stain and negative stain, which you guys all should have done before the storm. Yes?
Okay, so on Monday,
Yes.
a plate and then a hydrating steel stain, is that all we're doing Monday?
And we're going to be doing, for Monday, we're going to be doing, your TA will do a little review with you guys, and then you guys will also vote on the auger art for which one's best. Very good. And then Wednesday is when you guys will have your lab practical. Oh, and real quick, for those of you who are OLA students, I'm going to send out a message to you guys in just a minute, but we have a separate thing set up so that you guys are able to take it, being able to view the different types of media. Yeah, that's a really good question.
So then on Wednesday, there's 25 stations.
Yes.
Is the written portion, and are we analyzing what we did on Monday, or?
No, there'll be things that I set up for you guys. Yeah, absolutely. Any other questions? Does anybody have any blanks they'd like me to fill in or help them fill in with the sheet? Okay, if you guys have any questions, come up and see me. I'll chat with you guys for a bit. You guys are done. End of lab lecture.